Hello, my name is Azim,
My name is Yash.
And my name is Akash.
and today we will be introducing a novel vacuum-sealed injection system, helping restore skin health for astronauts in space with our novel biology and engineering. Our design was inspired by our time at NASA's Gene Labs as bioinformatics research interns. As for our contents, the labels are shown here below, and you can follow along on the left side. Without further ado, let's get started.
Our project analyzed RNA sequencing data from NASA's OSD-254 dataset, focusing on dorsal skin samples from 10 mice, 5 in ground control and 5 in spaceflight, all euthanized at 75 days. The slide shows a meta-analysis of the dataset. Using Galaxy, we identified genes and pathways altered between ground control and spaceflight. The plot shows up and down regulated genes, with the 3 circled cell cycle genes downregulated but potentially stabilized by IGF2. Our GOSEQ shows a strong link to cell cycle regulation, with significant downregulation in space. ShinyGo showed these genes are interconnected with IGF2, sharing cell growth functions.
To find IGF2's relationship with the cell cycle genes, we found pathways they shared through literature, starting with IGF2's role as a cell receptor and following the processes it's involved in. We found the MAPK pathway to be a crucial mediator. On the bottom left, we characterized this pathway, where we found it to be responsible for taking in signals like stress, like microgravity, and causing changes in cell proliferation and differentiation. On the top right, we have a biological mechanism IGF2 uses to increase cell cycle gene expression. You can take a closer look at it by pausing the video. Now on the left, we have a study that shows IGF2's impacts on structural proliferative units in skin. Essentially, researchers found that mice with higher IGF2 doses increase the fraction of dividing cells in overall tissue area. This means IGF2 promotes skin thickness and better skin health. On the right side, we visualize the impacts of IGF2 in relation with the cell cycle regulation genes, where it impacts cell proliferation, skin thickness, and wound healing functions. Now, merging our novel biology and engineering, let's talk about what we aim to address with our design and what our plan with IGF2 actually is. First, IGF2 will be housed in lightweight radiation-shielded pods that maintain a stable vacuum, preventing air injection and protecting from radiation. Next, our IGF2 storage solution includes trehalose, polysorbate 20, and lipoprotectants to protect against temperature fluctuations, thus preserving protein integrity. Lastly, our syringe design with single-use needles and channels will ensure accurate subcutaneous delivery of IGF2.
The IGF2 injection system is designed with key principles in mind: reliability, safety, or sterile drug administration while preventing air injection and protecting IGF2 in space, efficiency, or mass and volume reduction, and accessibility. We've also optimized our design to minimize air embolisms, as seen in these fluid simulations demonstrating the efficacy of the vacuum seal against comparable solutions. The IGF2 injection system is constructed of a body tube of CNC stainless steel, rubber gaskets, protective foil, and PTFE membrane. This is in stark contrast to expensive equipment administering drugs currently, and by utilizing separate, lightweight drug capsules, we can save mass and volume. This design can be manufactured at scale, with small costs when producing in bulk. On the bottom right, we have a short breakdown of the materials cost analysis. Here's our design functioning through an animation. You can see the simple swap-in, swap-out architecture of the system, allowing reuse of the main body tube while maintaining sterility by disposing of pieces that will enter the skin or contact the medicine. On the side are physical 3D printed prototypes of the system.
Our IGF2 injection system restores disrupted molecular pathways to counter microgravity-induced skin deterioration. Unlike topical treatments, it ensures deep tissue penetration and lasting effects. Vacuum-sealed pods cut weight, cost, and contamination while preserving drug stability. The syringe system will be applicable both terrestrially and in space, potentially for all compatible liquid medications, thus benefiting astronauts and having applications for both burn victims and dermatology. As space medicine advances, our solution will be both innovative and commercially viable. To conclude, our IGF2 injection system restores cellular function, ensuring astronauts' skin health in space while addressing a $1.6 billion market on Earth. Designed for FDA and spaceflight compliance, we are conducting microgravity simulations and preclinical testing with NASA and Odyssey Spaceworks. By advancing space drug delivery and regenerative medicine, we are not just innovating, we are securing the future of human exploration.
With that, thank you so much for joining us, and we look forward to continuing our work, advancing space and terrestrial medicine.